So today we are going to talk about the decidua. Decidua is nothing but the uterine lining, okay, endometrial lining which will shed during parturition. And here, before going to the decidua proper, you should know what is called as chorionic plate. Initially, everybody knows about the zygote formation and the blastocyst, blastomere, everything. Now, the next stage of blastocyst, blastomere is the cells which are surrounding the embryo is called tropoblastic cells, which are present inside this embryonic pole and abembryonic pole. So this cells start to see, first one is male gamete, female gamete together forms a zygote. The zygote will be getting multiplied and 16 cell stage, 32 cell stage, and then the fluid, uterine fluid enters into the blastomere and they pushes the cells towards periphery and they form a blastocyst. Now what happens? Now this pole is called as embryonic pole. So one pole, the embryo is there, that is called as embryonic pole, and the other pole is called as abembryonic pole. So the embryonic pole, the cells are two cells. One is called as embryonic pole. These cells will be transformed into embryo. The rest of the cell which lines it is called as trophoblast. These trophoblast cells will be multiplying and forms a villi. Initially, you will be having the primary villi, secondary villi, and tertiary villi. This chorionic villi is formed from a plate. That plate is called as chorionic plate, and this plate, the villi are extending into the uterine wall. The uterine endometrium which is receiving the villi is called as decidua. The uterine endometrium which receives the villi is called decidua. Now what happens, you can see this is the chorionic plate and this is the decidua, the uterine endometrium. Now see, look at the villi, it is entering here. The feto-maternal circulation happens here, and then now what happens, the villi which is adherent to the endometrium of the uterus, this area, the chorionic villi is very strong and bushy. This is called as bushy chorion. See, chorion frontosome, that is adherent. The opposite pole, it is not needed because it is not adherent to anything, because it is tightly attached with the chorionic plate. Since it is bushy and it is called as chorion frontosome, this area, the decidua is thick and it is called decidua basalis, which is shed during partition, and it contains decidual plate and lipid and glycogen. The next layer, abembryonic, opposite to the embryo, which covers the embryo, it is called as decidua capsularis. Then the third layer which lines the other part of the uterine cavity is called decidua parietalis. Now what happens, when the embryo is growing up, this chorion leaf, this smooth part, it is called smooth portion, and this is called bushy chorion. It is called as chorion frontosome and it is called chorion leaf, smooth part. This will, when the embryo is growing, this will come and it will merge with the decidua parietalis. The uterine cavity is full. So this is very important about the decidua. So there is a decidua basalis. You have to add one extra point as decidua capsularis. This layer becomes stretched and degenerates. For the decidua basalis, you have to write this contains lipid and glycogen. And then last, only which portion of the embryo takes place in the gases of transportation of the gases and nutrients is chorion frontosome. Since it is attached with the uterine wall, here only the nutrients transportation takes place. So that's all you can write about the decidua. So then future, the decidua basalis make up together with the chorion frontosome, which the decidua basalis makes up placenta. That's all about the short notes for decidua. You have to write one diagram, how to draw. Start small embryo, you have to write a small embryo. So the embryo is covered by the amnion. The next covering is the chorion. While drawing the chorion only, you have to draw carefully. Here you have to draw the bushy chorion frontosome, bushy chorion. When you draw the bushy chorion, you have to draw the decidua basalis, the next layer. So this is the uterine wall, you have to draw. So here I will use separate color because decidua is to be drawn in the separate color. So this is the decidua which is attached to the chorion frontosome. This is decidua basalis, and then you cover this area. So this is covering the embryo, no, right? This is chorion leaf, Decidua capsularis. Then this is Decidua, inside you have to draw Decidua parietalis, rest of the rays. So while drawing, you have to draw like this. Slowly, when the embryo is going big, this layer, entire layer will be obliterated. So while marking the parts, you should mark this as embryo. So you put an I. Embryo, this is the amniotic cavity, one which is lining here. Next is the chorionic cavity. So this is the chorion frontosome, bushy chorion frontosome or bushy chorion. And this one is the chorion leaf or smooth chorion. And then next is the Decidua, which is adherent to this, is Decidua basalis, which contains lipids and glycogen, and this contains this is Decidua capsularis, and this is called Decidua parietalis. And when the embryo is growing up, this Decidua capsularis comes and merge with the parietalis and closes the uterine cavity. That's all about Decidua. In Decidua, in future, in this part of the Decidua will be transformed into placenta, which is important in the transportation of nutrients and gases.